Morning students, today we are going to study about the female reproductive part, gynoecium. Gynoecium, it includes two or more pistil. Here pistil means it includes ovary, style and stigma. Here stigma, it is the platform for the pollen grains. Next style, it's a connecting tube for stigma and the ovary through which only the pollen tube travel and reaches the embryo sac for fertilization. Next one, the basal part. This is ovary. It includes ovule. Ovule is otherwise called megasporangium. Ovule is otherwise called megasporangium. Let's see the parts one by one. This is chalazal end. This is integument, nucellus, embryo sac, raphe. This part hilum. Then this gap micropyle. Then this is funicle. Let's see one by one. The basal part, this is an inverted ovule. The basal part where nucellus, integument and funicle all will merge, that is chalazal end. Next one, the central part. The central part of the ovule is nucellus. It consists of the food material for embryo sac. Here it is covered by a layer, protective layer, that is called integument, and it is leaving a gap. That gap is called micropyle. If it contains only one layer, means it is called unitegmic. If it contains two integument, we can call that as a bitegmic ovule. Next, the stalk of the ovule is funicle. In that only attaches the ovule to the ovary by the tissue placenta. Then the point of attachment of the ovule and funicle is hilum. Next one, the oval shaped structure found at the micropylar end of the nucellus is called embryo sac. It is otherwise called as the female gametophyte. It consists of 7 cells and 8 nucleus structure. Here it is surrounded by nucellus. In some plants, the inner layer of integument may become specialized and perform nutritive function for the embryo sac. Example, Asteraceae. That type of integument is called endothelium. That is called endothelium. Next, hypostasis, a group of cells found at the base of ovule. This part, chalazal and embryo sac. This is called hypostasis. Next, epistasis means above, above. Thick-walled cells above the micropylar end as well as the above the embryo sac. That is epistasis. Hypo means base, epi means above. Next one, types of ovule based on the sporogenous cell. Here the sporogenous cell, if it is hypodermal, we can call that as a tenunucellate. If it is subhypodermal, we can call that as a crassinucellate. Among these two types, which one have more amount of this nucellus means it is crassinucellate. But tenunucellate have very small nucellus. Next we can see the types of ovule based on the orientation, shape. Types of ovule. There are 6 types of ovule based on the orientation, form and position of the micropyle and funicle and salasa. So there are 6 types of ovule based on the position of micropyle, funicle and chalasa. Let's see the 6 types. First one orthotrophous, second one anatrophous, third one chemianatrophous, fourth one combylotrophous, fifth one amphitrophous, sixth one cecinotrophous. The main types of ovules are ortho, ana, hemi and combylotrophous. First one orthotrophous. Here the micropyle, the opening is at the distal end, then the micropyle, then the chalazal part as well as the funicle all at the same vertical line. This is orthotrophous. Example, Piperaceae, Polygonaceae. Next one anatrophous. This is an inverted type. The body of the ovule is completely inverted. So the micropyle as well as the funicle both lie very close to each other. Micropyle, funicle close to each other. Here this type is common in dicot and monocots. Next one, hemi-anatropous. Here the body of the ovule is placed at the right angle to the funicle. The body of the ovule is placed at the right angle to the funicle. Example, Primulaceae. Hemi-anatropous. Next one, Compylotropous. Here the body of the ovule is curved in the micropylar part. The body of the ovule is curved at the micropylar part, more or less bean shaped. More or less bean shaped. Example, Leguminaceae. So the main types of ovule, if all in a vertical line, micropyle, chalaza and funicle mean orthotropous. If it is inverted, that is anatropous. Body of the ovule is at right angle to the funicle mean hemi-anatropous. If it is bean shaped mean compylotropous. Some other two types also will be there. Let's see that. First one, amphitropous. Here the distance between hilum, chalaza is less. So it will look like a horseshoe shape. So the horseshoe shaped is amphitropous. Example, some Alismatiaceae. The next one, Cercinotropous. Here the funicle is very long and it surrounds the ovule. The funicle is very long and it surrounds the ovule. Example, Cactaceae. So if it is horseshoe shaped, the ovule is horseshoe shaped means that is amphitropous. The funicle is very long means that is Cercinotropous. Next we can see the female gametophyte. Megasporogenesis. The process of development of megaspore from a megaspore mother cell is called megasporogenesis. You know very well about microsporogenesis. Here in the female gametophyte, it is a megasporogenesis. See, first central part of the ovule, that is nucellus. It's nothing other than a group of parenchymatous cells. In that, one cell will become specialized and it will function as a archisporial cell. This archisporial cell, in some plant, it will directly function as a megaspore mother cell, but in some plants, it will divide into parietal cell and sporogenous cell, like the male gametophyte. That parietal cell undivide, but the sporogenous cell divide repeatedly and it will produce sporogenous tissue. Last generation, megaspore mother cell. Then this megaspore mother cell undergo meiotic division and it will form 4 megaspores. 4 megaspores. Among the 4, the one which is facing the chalazal end will become a functional megaspore. So based on the number of megaspore developing the embryo sac, we can have 3 type of embryo sac. One is monosporic, second one bisporic, third one tetrasporic. If only one megaspore, the chalazal end facing one, developing into embryo sac mean we can call that as a monosporic. Second one, bisporic. If 2 megaspores are developing embryo sac means we can call that as a bisporic. All 4 megaspores are developing embryo sac means we can call that as a tetrasporic. Example, peperomia. Bisporic, allium. Monosporic, polygonium. Then how the embryo sac will be developed? Let's see. See here the functional megaspore megaspore. It will undergo mitotic division and it will result in the 2 nucleus. Then this 2 nucleus again divide mitotically and it will result in the 4 nucleus. Then this 4 again divide mitotically and it will result in the 8 nucleus condition. Then how that 8 nucleus will organize? Let's see. See the 4 nucleus towards micropylar end. The 3 will become a egg apparatus. The 3 will become a egg apparatus. 1 will be freely in the cytoplasm, that is upper polar nucleus. Among the 3 towards the micropylar end, the 2 are synergids and the 1 is egg. Next, the 4 nucleus towards the chalazal end, the 3 will become antipodal, then the 1 will be freely in the cytoplasm, that is the lower polar nucleus. See, in some plants the 2 polar nucleus remain free. In some plants the 2 polar nucleus join together and it will become a secondary nucleus. Then this filiform apparatus, it is helping for pollen tube entering into the egg as well as it is helping for conduction and absorption from the nucellus to embryo sac. And the synergids, it is secreting some chemical substance for the pollen tube reaching the egg. Pollen tube reaching the egg. So filiform apparatus helping for the pollen tube entering into the egg. Then the synergids also helping by secreting the chemical. Then the two polar nucleus at the center. In some plant both are joined together and it will become a secondary nucleus. Then the three will be at the chalazal end, that is called antipodal. Now the embryo sac is 7-celled and 8 nucleus structure. 7-celled and 8 nucleus structure. Thank you.